Students, this is our next lecture from chapter Biomolecules, and now in this lecture we will learn about our next topic, enzymes. Now see on the screen you are looking the topic enzyme. There are many kinds of chemical reactions which are takes place at a given time in the cells. You all are also familiar about the chemical reactions which are taking place in our body, in plants or in each living cell. So these reactions are takes place at body temperature. This just because of an enzymes. So enzymes are nothing but these are the biocatalysts which are made up from the proteins. Now see, a German chemist, Edward Buchner, discovered enzyme by accident. Buchner discovered that living cells were not necessary but that yeast extract could bring about fermentation. He then coined the term enzymes, which means Greek word en means in, Greek word zyma means yeast. That is, in yeast, this term is now commonly used for all biocatalysts. The substance on which enzymes act is called substrate. The enzymes which acts within cell called endoenzymes. For example, enzymes of chloroplast and mitochondria. And if they acts outside the cell are called as exoenzymes. For example, enzymes of fungi. These fungi are secreting enzymes and acting outside the cells. Such kind of enzymes retain their property, their catalytic property, even when extracted from cells. Now see on the screen the tablet rennet used for coagulating milk protein, that is casein, in cheese contain rennin enzyme. That rennin enzyme is obtained from stomach of calf. Chemical composition of enzymes. Now we will see. So there are on the basis of chemical composition, enzymes are of two categories. Now, number one, purely proteinaceous enzyme, for example, proteases, which are made up from proteins. Enzyme protease acts on proteins. Now, number two, conjugated enzymes. These conjugated enzymes are made up of proteins and also from non-protein prosthetic group is attached to them firmly by chemical bonds, due to which it is not removed on hydrolysis, and if it is removed then enzymes become inactive. So these are the two kinds of enzymes, purely proteinaceous enzymes and conjugated enzymes. and we have seen the nature of enzymes. So enzymes, if we try to break protein part and non-protein part from conjugated enzymes, then enzymes will become inactive. Now we will see some terms. That terms are: number one, coenzymes. Coenzymes are organic compounds tightly attached to protein part. For example, NAD, nicotinamide adenine dinucleotide, or FMN, flavine mononucleotide. So these are the some examples of coenzymes. Next, cofactors. These are the inorganic ions which are loosely attached to protein part. For example, enzyme peptidases shows magnesium, zinc ion, copper, etc. are inorganic ions which forms cofactors. Now next we will see about properties of enzymes. Which are the properties which are shown by enzymes? Number one, they have proteinaceous nature, means all enzymes basically made up from proteins. Number two, they shows three-dimensional conformation. All enzymes have specific three-dimensional conformation, shows one or more active sites for binding to substrate, which is also called as substrate binding site. Number three, they shows catalytic property. Enzymes are like inorganic catalyst which affects the speed of reactions without changing themselves, and again ready to act on a next substrate by producing desirable product. A small quantity of enzyme can catalyze a large quantity of substrate into product. For example, substrate sucrase, enzyme sucrase can hydrolyze 1 lakh times of sucrose as compared with its own weight. So this is the catalytic property of enzyme. Next property, specificity on action. Enzymes show specificity on action. Enzymes show specificity to act on specific substrate only. Number fifth, reversibility of action. Enzymes are very sensitive to temperature and pH. Each enzyme shows its highest activity at specific pH and temperature, called as optimum pH or optimum temperature. Any decrease or increase in pH or temperature changes the rate of reactions. For example, enzyme pepsin, which is present in stomach, shows highest activity at pH 2, means at acidic pH, whereas trypsin, which is found in duodenum, which acts at 9.5 pH, means at alkaline pH. Both digest proteins into amino acids. Enzymes are destroyed at high temperature up to 60 to 70 degrees centigrade or below also, but can again become active at suitable temperature. Most of enzymes work at an optimum temperature. That temperature is between 20 to 35 degrees centigrade. So these are the some properties of enzymes. Now our next topic is nomenclature of enzymes. There are many ways to naming enzymes. Enzymes are named by adding suffix ase, ase, to the name of substrate on which they act. For example, protease, nuclease, sucrase, etc. Enzymes also named according to type of function. For example, dehydrogenase removes hydrogen. Oxidase In oxidation, decarboxylase removes CO2. Some enzymes are also according to source from which they obtain. They are named as papain, from papaya, bromelain, from members of family Bromeliaceae. According to International Code of Enzyme Nomenclature, each enzyme ends with ASE or ACE and consists of double name. The first name indicates the nature of substrate and second indicates reaction catalyzed. For example, pyruvic decarboxylase, which catalyzes and removes CO2 from pyruvic acid. So these are the some ways on the basis of which we are classifying enzymes. Now in detail we will see the classification of enzyme. Number 1, oxidoreductase. See on the screen, these are the enzymes catalyzing oxidation and reduction reaction, means redox reaction, by the transfer of hydrogen or oxygen. For example, alcohol dehydrogenase. Now see the reaction, alcohol plus nicotinamide adenine dinucleotide with the help of alcohol dehydrogenase, which produces aldehyde plus NADH2. So these are the oxidoreductases. Number 2, transferases. These enzymes catalyze the transfer of certain groups between two molecules. For example, glucokinase. Glucose plus ATP. In presence of glucokinase, it forms glucose 6-phosphate plus ADP. See next, hydrolases. These are the enzymes catalyze hydrolytic reactions. This class includes amylases, proteases, lipases, etc. For example, sucrase. Sucrose, in presence of enzyme sucrase, forms glucose plus fructose. Next, lyases. These enzymes are involved in elimination reactions resulting in the removal of group of atoms from substrate molecules to leave a double bond. It includes aldolases, decarboxylase and dehydratases. For example, fumarate hydratase. See the reaction histidine in presence of histidine decarboxylase, which removes CO2 and forms histamine. Next, isomerases. These enzymes catalyze structural rearrangements within a molecule. Their nomenclature is based on the type of isomerism. Thus, these enzymes are identified as racemases, epimerases, isomerases, mutases, etc. For example, xylose isomerases. Glucose 6-phosphate in presence of isomerases form fructose 6-phosphate. So these are the examples of isomerases. Next are ligases or synthetases. These are the enzymes which catalyze the covalent linkage of molecules utilizing the energy obtained from hydrolysis of an energy-rich compound like ATP, GTP. For example, glutathione synthetase, pyruvate carboxylase. See the reaction, pyruvate plus U2 plus ATP in presence of pyruvate carboxylase forms oxaloacetate OAA plus ADP and inorganic phosphate. So this is the classification of enzymes. Now on the screen you are looking some questions. You have to solve these questions. Number 1, which enzyme is needed to digest food reserve in castor seed? Options are there. Number 2, coenzyme is, we have discussed about coenzyme, cofactors, so definitely we'll see answer there. Number 3, list the important properties of enzymes. Number 4, name the chemical found in the living cell which has necessary message for the production of all enzymes required by it. So these are the some questions you have to solve, and if getting any problem, then contact to your subject teacher. Thank you.